Hi, I'm Cameron Burke, and welcome to this Kaplan MCAT Science Review for the innate immune system. This might seem like an intimidatingly long list of topics, but it can actually be broken down into only 3 categories that we need to review, including a general overview of innate immunity and what that is, a general description of the molecular innate immunity barriers your body has, and finally, we'll cover your cell-mediated responses and the different cells involved in innate immunity.
When you're through with this video, ideally you'll have a general understanding of what the components of the non-specific immune system are and how they work individually and as a team to keep you healthy. This goal should help you answer questions like this one about the role mast cells play in immunity. As a broad term, immunity is the ability of an organism to resist disease, but there are different types of immunity we have at our disposal to fight infections. The first type of immunity is innate immunity, sometimes called non-specific immunity, which resists any kind of foreign invader. And the second type is adaptive immunity, which responds by recognizing specific microbes and fighting them individually. The star of this particular video is innate immunity, so don't worry about adaptive immunity so much at this particular second. There are 3 tiers of innate immunity that separate you from all the gross stuff in the world. The first are physical and chemical barriers, like skin and tears. The second is specialized proteins that fight infections. And finally, you also have immune-related cell-mediated responses. Keep this basic organization in mind as we get into more detail about each of these types of immunity throughout this video. The first resistance most germs encounter are physical and chemical barriers that don't have to be activated. They're always there, just waiting for infections to try and get in. Tears and saliva are one example of this phenomenon. Both have lysozymes that can burn up bacteria and viruses, so while it might seem like your tears are just sitting there doing nothing, they're actually killing invaders coming in through your eyes. Lysozymes chemically destroy invading organisms, but you also have more humble barriers like mucus secretions and cilia. Mucus basically just gums up the works so bacteria get stuck, and then cilia push them out of your body. Should organisms make it past your tears, saliva, and lung mucus, your stomach is the next line of defense. In case a foreign organism makes it past your mouth, the contents of your stomach has a pH of approximately 3 and is waiting to dissolve the invading organisms in acid. Although it lacks the dissolving power of hydrochloric stomach acid, your skin is the most formidable physical barrier you have in terms of size. It's the single largest immune defense. In addition to just physically separating the inside of your body from everything else in the world, it also has defensins to kill unnecessary bacteria. On occasion, barriers don't get the job done, but once foreign organisms get into your body, you have interior lines of defense as well. Complement proteins are one of these defenses. Generally, complement proteins float around in your blood and punch holes into bacterial cell walls to mess up bacterial ion gradients and thus kill the bacteria. There are 2 ways complement proteins can be activated. The first is the alternative pathway, which doesn't require antibodies and is an example of non-specific defense because it does not discriminate between different types of foreign organisms. It's called alternative because antibodies are not involved, and typically you think of antibodies when you think of immunity. But in this video, our focus is on non-specific immunity, so for our purposes right now, the alternative pathway is actually the more important pathway. The second pathway is the classical pathway, which does use antibodies bound to pathogens to assist in destroying the pathogens. But this is also an example of non-specific immunity because antibodies are not modified to be more likely to target one type of pathogen over other types. There just happened to be a generally applicable antibody involved in finding foreign cell walls. While complement proteins are good for dealing with bacteria, viruses don't have cell walls, so they need different proteins to stop them. Interferons are the proteins in charge of preventing viral replication. Usually the virus perforates a cell, manipulates the cell's machinery to replicate the virus, and then leaves the cell to infect others nearby. Interferons limit all of these processes. Firstly, interferons limit how permeable the cell is, which stops viruses from accessing tools for replication before they can even get started. Secondly, interferons slow down regular protein production in nearby cells. So in the event the viruses do access cellular machinery, the machinery isn't producing proteins of any kind, so the virus has a tougher time reproducing and moving on to find new cells to infect. Interferons basically stop new little viruses from being born, so there's nothing left to leave the infected cells, and the infection's contained. Interferons are actually part of what makes you feel sick. So when you're super tired and have a headache and a face full of snot, that actually means your immune system is doing its job. A bigger concern would be if you were infected with a virus and you still felt fine, because that would mean your interferons were asleep at the wheel and not containing any viruses. Complement proteins and interferons are really important backups for your physical barriers, but they're definitely not the only interior response you have to being infiltrated by pathogens. In addition to protein responses, there are cell-mediated defenses you could be asked about on test day. When pathogens get past your physical barriers, your cell-mediated innate immune system gets on the scene. Firstly, wherever a pathogen happens to break the barrier becomes what basically amounts to an immune system crime scene. The site gets inflamed and the innate immune system sounds the alarm to get a wide variety of immune-related cells to the area to get ready to do their thing. We'll get into those in more detail shortly. The second thing is that phagocytes, or white blood cells, just start straight up eating any foreign cells present. Finally, antigen-presenting cells pick up the dead bodies of the invading pathogens and hold them out so the foreign proteins are recognized by T cells and acquired immunity can be fostered. Inflammation is the first step of the innate immune system because it basically does the job of a first response team at the scene of an accident. Inflammation makes an immediate area less hospitable for pathogens by increasing heat and blood flow to bring immune cells and clotting factors to kill present pathogens and stop more from getting in. It also is the first to ring the alarm to the immune system that something's gone wrong, initiating immune response. In addition to alerting the immune system that something's up, swelling literally makes it more difficult for pathogens to both enter the body and move around once they're in. Heat can also kill bacteria. Inflammation begins when cells that have been injured or are dying, or immune cells who happen to be in the area, send out chemical messages to the rest of the body that they're in trouble. The big category of these types of cells to worry about include mast cells, macrophages, and dendritic cells. Following inflammation, phagocytosis is the second broad category of cell-mediated response. Macrophages are basically big white blood cells that swim around your blood looking for a bacterium, which can be located by the pathogenic peptides that act as antigens in the bacterial cell walls, which are then targeted by antibodies marking them for destruction. Macrophages just basically eat bacteria Pac-Man style, and then that's the end of that pathogen. It's important that macrophages eat bacteria, but what they do afterwards is even more important. Once a bacterium has been broken down in a macrophage, major histocompatibility factor binds the broken down antigens and carries them to the surface. That sounds very scientific and neutral, but what's actually happening is that the macrophage eats a bacterium, then dissolves it, and then shows the rest of the bacteria who's boss by holding up dissolved bacteria body parts to show the rest of the immune system what they should look for, activating specific immunity. You would think that would be scary enough to make the rest of the bacteria just flee for their lives, but if that doesn't happen, the displayed antigens allow adaptive immune system cells to know what to look for, and then to locate still living bacteria in the blood and lymph and get rid of those pathogens. As a general category, macrophages are responsible for eating invaders, but knowing where they come from is something you might have to be responsible for on test day. Macrophages are agranulocytes, which don't have cytoplasmic granules. We'll review granule-having immune cells shortly. These guys come from monocytes, which are blood-borne. Monocytes are basically little harmless baby cells who just float around until they're triggered to turn into macrophage eating machines. This usually happens when bacteria enter the tissue, resulting in a necessary immune response. Once monocytes get turned into macrophages, they're part of the cellular team that turns on inflammation and recruits other immune cells to the point of entry. As you'll recall from a second ago, major histocompatibility factor is super important for macrophages, but it's actually present in all nucleated cells and is used by the immune system to monitor the general health and functioning of cells. MHC class I molecules operate on what's called an endogenous pathway, because it works from the inside out, binding antigens after they're already in the cell. For example, viral molecules that have entered the cell get broken down into their antigenic peptides. Class I MHC molecules pick up these peptides, and then the peptide attached to the class I MHC molecule are moved to the surface of the cell for the immune system to check out and act upon if necessary. Because MHC class I cells aren't just for immunity, they have to do regular cell tasks too, and that includes breaking down used up proteins from cellular activities in the same way that pathogenic proteins are broken down. All broken down proteins go to the cell surface. Any non-self proteins that appear on the surface trigger an immune response, while self proteins that appear on the surface don't trigger anything. All nucleated cells have some sort of MHC molecules, but class II MHC molecules are usually found in cells whose only job is immune-related. Unlike MHC class I, MHC class II works on an exogenous pathway, because it attacks antigens that come directly from outside of the cell, instead of from being broken down on the inside. That said, they still end up displaying the antigen peptides on the surface of the cell to trigger both innate and adaptive immune responses. So far, the cells we've reviewed are the first things the immune system tries when trying to contain and eliminate an infection. Natural killer cells, as you may be able to guess from their name, are not the first resort. They're the immune system's big guns for when the first wave of defenses don't work, and a virus manages to evade detection from B-cell responses. Natural killer cells get called in after a virus invades a cell, and the virus gets control of MHC molecules and turns them off before the pathogenic peptides can be displayed. So T cells can't see that there's an infection just by looking at the surface of the attacked cell. Natural killer cells, however, can sense the downregulation of MHC molecules, and then they respond by forcing the cell to kill itself, taking one for the team by forcing the virus to die with it. Forcing otherwise healthy cells to kill themselves isn't a great first choice, so this only happens when other immune responses don't get rid of the infection inside the cell first. Think back to a couple moments ago when we reviewed agranulocyte macrophages. Those cells were just responsible for eating and baiting pathogens, but they also have granulocyte counterparts who play a variety of important immune roles. The three big categories to worry about for the MCAT include neutrophils, eosinophils, and basophils. All of these have little granules in their cytoplasm that can be seen with a microscope. And a good rule of thumb is that granulocytes in general are involved in inflammation response. The first of our granulocytes to review are neutrophils, which are white blood cells responsible for using their short little lives to hunt down and eat bacteria. They can locate bacteria using chemotaxis, or following a chemical trail, which is left by opsonized bacteria. Opsonized is just a technical way of saying the bacteria have been tagged by an antibody, which is what the neutrophil picks up on. Unlike neutrophils, eosinophils don't really eat things. Their job is to release histamine to increase inflammation when foreign bodies like parasites such as worms breach barriers. Histamines basically trigger an inflammation response, and eosinophils are generally identifiable by their bright red granules. Eosinophils allow immune cells to move out of the bloodstream and into surrounding interstitial space at the site of the injured tissues by the point of entry. While eosinophils trigger inflammation due to injury and parasites, basophils and mast cells are in charge of releasing histamine in response to allergens. So next time you have an immune response to cats or flowers, you can blame these guys. Basophils are identifiable by their big purple granules. Mast cells have slightly smaller granules and are primarily present in tissues exposed to the outside world, generally in places where you find mucosa and epithelium. Under normal conditions, these guys are present in the smallest numbers, only being produced in bulk when their triggers are present. Now that we've gone through all the content for this review, you can check out your learning goal progress with some practice questions. The first one deals with the role mast cells play. The correct answer is B. Mast cells release histamine in response to allergens. The second practice question tests you on complement proteins. If you pick D, you've got the right answer. The classic complement protein pathway involves antibodies bound to pathogens. Last practice question. This one tests your knowledge of the immune response to parasites. A is the correct answer. Eosinophils are the main cells in charge of responding to parasitic infiltration. To recap, you have many innate immune responses, including macrophages, which eat pathogens, as well as mast cells and granulocytes, which each promote inflammation response to infections of different kinds. The innate immune system also relies on antigen-presenting dendritic cells to show T cells what kind of proteins need to be destroyed, and natural killer cells, who are the last resort big guns, who destroy infected cells or cancerous cells before the disease can spread to other parts of the body.
And that's the end of the innate immune system review. Hopefully, if you see questions about the innate immune system on the MCAT, you'll be able to show it who's boss.